Well, I have to say that this is the third time that we're doing the Triangle Biotech Research Symposium, and it's my second time being here. And I was able to come in early enough yesterday to participate in a lunchtime program that the COIN group organized. You're going to hear from Joe Magno in a moment. And it's just so lovely to feel so welcomed and to just really have a lovely experience. And thank you. And what gorgeous weather is this? We're based in New Jersey, where it's a nice 65 and sunny most days when we're lucky, but it's not 80 at this time of year. The Biopharma Research Council is about 5 years old, almost 6 years old. And essentially, we are an organization designed to bring people together, scientists together, across all of the different functional silos. So that is industry, academia, nonprofit, government, and supplier labs, and the people who love them. Basically everywhere that PhDs work. And what we try to do with each of our different educational programs is to either have a clear content focus, but sometimes it's a strategic focus like today. We're going to be talking all day long about partnerships and collaborations. And what I appreciate about today is that we have a number of examples of successful collaborations. And I was just commenting to someone earlier, you know, if you look back the last 5 years, many, many stellar collaborations have been announced, and then you never hear from them again. And so I am very appreciative that we have some folks today that are going to talk about their successes, their lessons learned, and so on. Let's see, I was taught how to use this. It works. Well, we have a lot of people to thank. First of all, I thank all of you for being here. We have a wonderful committee, which is on the back of your handout. This is the first time that we've had a committee working with us, and you can see it's a really diverse group. And yes, if you're interested, talk to me, because we'll be forming our committee for next time, which is likely to be next August. Please take some time while you're here today to speak to the different sponsors. They're the ones who make your food possible, for sure. The only other remark I wanted to make, I mean, what I appreciate about our organization is that it's so much about serendipity. And one of my favorite notes I read recently, so my kids gave me a birthday book that is all about the origins of things. And my big takeaway was that it was 3,500 years between the sundial and the pocket watch. All right? Now, there's a clock in everything we use. There's a camera in everything we use. There's a microphone in everything we use. And what's next, right? So everyone here I know has been working on important programs for many years, some short, some longer than others, and are constantly generating new programs. So thank you so much for being here. We have a couple of our committee members who are going to make very brief remarks about what they do, starting with Winona Satcher from the City of Durham.
Thanks, Diane. Good morning, everyone. So my name is Winona Satcher, and I work for the City of Durham, North Carolina, and founder of the Durham Urban Innovation Center, or DUIC. Welcome to the third Biotech Symposium. I'm here to help bridge the gap between biotech and civic spheres, STEM clusters and community engagement, and research to practical application in low-wealth communities. I'm excited to be here to further the discussion around your work and its potential for positive impact at the neighborhood level. As a start, my colleague Robin Dixon, whom some of you may have met this morning passing out postcards on the side of the registration table, and I will continue to do that and inviting you as special guests to our Spring 2015 Job and Youth Learning Fair. We hope to pair our young leaders with your organizations in efforts to develop hidden talents and attract those unforeseen future and current science leaders. I also hope to partner with you and your organizations to continue that effort and to create innovative, community-wide, scalable STEM initiatives in the Triangle region. The DUIC, or the Urban Innovation Center, works collaboratively with residents and stakeholders to solve urban challenges. I believe this Biotech Research Symposium is a wonderful platform to do that. So I want to thank Joanne for asking me to be on the planning committee. It's been great. So I'd say to all of you to get your filled with coffee. I got to get my coffee soon, so I'm kind of like jittery. But enjoy the rest of your day. And again, I look forward to meeting with all of you, having some wonderful dialogue outside of the box, thinking how we can bridge the gap between what you do and what city officials do and the local communities that surround Research Triangle Park. Feel free to ask me any questions, Robin any questions throughout the day. And again, I look forward to working with you and hopefully partnering with you in the future. Thanks.
Good morning, everyone. My name is Joe Magno, and I'm the Executive Director of COIN, which used to be known to many of you as the North Carolina Center of Innovation for Nanobiotechnology. We recently changed our name to be reflective of the fact that we're really a network, and we work with folks like Joanne and the North Carolina Biotech Center to foster collaboration, particularly in areas where folks use tools and techniques of nanobiotechnology, which goes across a very wide range of topics and industries. So we're very fortunate to be here. We're very glad to be part of the planning committee and working with Joanne. This is all about everyone working together, networking, and maybe finding out something that you didn't know before. We're a 501c3 not-for-profit organization. We're membership driven. We're encouraging everyone to become members, of course. And we set up and put on events such as this and participate in events such as this just about every month or every few weeks to foster more collaboration and networking. So I'll be here all day. Please feel free to ask me questions and have a great conference. Thank you very much. Thank you, Joanne.
Okay. So our first panel, it's just a two-person panel, is about moving traditional drug development into the 21st century. Mark Parisi got in touch with us, interested in participating in our program, and I asked him, well, who are you working with? We need to hear that story as well. So he and Dr. Diora are going to speak together, and I'm going to ask you two to share the mic if you want to stand. Otherwise, you can sit at the table, but this way you'll be recorded, okay? Thanks very much.
Okay, so I hope you can all hear me. When I initially approached Joanne, I had fundamentally a scientific presentation in mind, which is usually about 30 slides with question and answer at the end. Joanne, as she has just stated, came back to me and said, well, this is all about collaboration. So let's possibly make it a bit shorter, bring in a collaborator, and do what you can to encourage audience participation. So I'm going to begin with a question. How many of you are involved in small molecule drug discovery? Okay. How many of you have some kind of interest in drug discovery? Okay. Okay, good. So I'm again Mark Parisi.
I'm Raj Nera.
Okay. So we're going to give some information about what we do individually, speak about our collaboration, but I would really like to encourage audience participation throughout the presentation we're going to give and not just at the end. So to give some background information, Astinex was founded in 1993. It was founded in Moscow, Russia. It's somewhat connected with the fall of the Soviet Union in that there were a lot of academic entities that were creating small molecules. And with lack of government funding, they really were unable to continue their research. So what we did at that point is we gathered these small molecules, created compound collections, and sold them in Western Europe, North America, and Japan. That went well. In 1996, we opened our own lab, again in Moscow, Russia. And we began creating our own compound collections, and we began orienting towards therapeutic indication. CNS, oncology, ion channels, kinases, et cetera. I could skip from roughly 1996 to 2013 because the big local development is that in March of 2013, we opened a lab in Winston-Salem, North Carolina. The compounds are now created there. They're QC'd, which mostly involves purity and confirmation of structure. We do some biological profiling and testing to prove our drug discovery orientation, and we have a compound management department too. Okay, so I would encourage questions. Would anyone like to question or comment at this point in the true collaborative spirit? Okay, so no one has had their cup of coffee yet? All right, we have more than 600,000 pharmaceutical precursors in stock. On a very regular basis, we work with collaborators in customizing compound sets to their specification. We also, again, test these compounds in-house to show and prove biological activity and therapeutic orientation. So briefly, I'm going to touch on 4 points. Our approach to library design, again, the orientation we have in particular therapeutic areas. We do not stop at the beginning of the discovery and development continuum. After initial screen, we provide a lot of follow-up service, okay, phrases that are well-known in drug discovery, hit the lead optimization chemistry, and people come to us throughout the discovery and development continuum with ideas about improving efficacy, solubility, permeability, repurposing, etc. So although our focus is on early drug discovery and an integrated set of products and services, at that point, we actually work with people during the entire process to bring a drug to market. We are chemists by training, so we do incorporate fairly sophisticated chemistry and really try to address underrepresented chemistry in screening collections many people have. And this allows us, or really gives us, the impetus to work on difficult targets in the area of protein-protein interactions. Another area we're very active in is the Wnt pathway, et cetera. So these are target areas that years ago were basically considered intractable, but now we are gaining some traction. Questions? Certainly?
So when you mention synthesis, are you speaking of organic synthesis, protein expression, or RNA interference?
Okay, when I speak about synthesis, I'm speaking about the creation of pharmaceutical precursors or small molecules.
Could be any of those.
Well, some of those things you just mentioned might be certain pathways or more on the biological side. Okay, we're kind of more in the traditional small molecule drug discovery area where we create compounds that are screened either in-house by our client looking for a hit, right? And then given when you have a hit, that's just a starting point, then you have to address improving efficacy. Sometimes there are solubility issues, unfortunately, permeability, toxicity. Okay, so this is kind of what we do. Good question?
Mark, so who is your market for?
Who is our market? Everything from a researcher at a university in a very small room with possibly one student working under his or her supervision to the biggest of big pharma and everything in between. Good questions. Thank you for making it an interactive session and not just me kind of going on and on. All right, to give a little more background information on the beginnings of drug discovery and compound collections, back in 1994 and really throughout the 90s, there was a combi-chem boom, and the idea behind combi-chem was, okay, through various synthetic methods, we can create more compounds, more in and more out. You have a basic hit rate if we create thousands or hundreds of thousands of compounds. And who can tell me what HTS stands for? Good. Okay, through high throughput screening and ultra high throughput screening methods, you can put in hundreds of thousands of compounds and you'll get more out, and hence you'll have a better starting point. That was basically disproven. Okay, who's this guy on the left? Okay, good. In 1997, he came up with the rule of 5. Christopher Lipinski, very, very famous. Okay, in drug discovery, this is one you have to know. Okay, who can tell me about Chris Lipinski? Anyway, he came up with the rule of 5, and he analyzed the number of compounds in clinical phases and on the market, and he said, look, let's come up with a starting point, okay, to address things like oral bioavailability and other issues, and kind of use these parameters in the compounds we create to begin with. And this kind of helped focus the CombiChem era, so people started creating better compounds from the beginning. Okay, so
What is the rule of 5?
the rule of 5 states that a number of parameters, molecular weight, hydrogen bond donors, hydrogen bond acceptors, C log P, et cetera, fall within certain parameters to allow a compound to go through discovery and development more quickly than other compounds that may have had issues based on these parameters not falling in with this umbrella concept, the rule of 5. Now, it's somewhat of a dated concept. It was 1997, and people are constantly speaking now about the adjusted rule of 5, the modified rule of 5, but really no one has come up with something to replace the rule of 5. It's been cited in maybe 1,500 papers or something like this. It's, you know, in drug discovery, Christopher Lipinski is very, very well known. And he worked at Pfizer, and I think it's during that period at Pfizer that he came up with this. But anyway, that's a while ago now. So the idea, given the rule of 5 and some other ideas in early drug discovery, was to come up with compounds from the beginning that are more lead-like, that are less likely to encounter some of the problems that maybe compounds in the past would have. Okay, and again, so quantity matters more than quality. This is the idea that quality is far more important. You create compounds at the beginning that are going to address things like solubility, permeability, maybe an orientation towards a particular target, et cetera. Okay, what we have done now is we've incorporated elements of nature into the compounds we create. In 2009, there was a very famous paper. Has anyone here heard of Escape from Flatland? Okay, good. We have someone here very well versed in drug discovery. Would you care to provide a few words? No? Okay. Okay, so in 2009, just 5 years ago, there were analyses done on compounds, again, the starting point for drug discovery in the market. And the idea was most compounds were flat. Very simply, it would be better to incorporate more chirality or make compounds more three-dimensional. And we've really worked under this umbrella concept now for 5 years. Okay, the chemistry is more sophisticated, but this is really kind of an underrepresented starting point in drug discovery. Would you like to add anything to that?
I think the focus on 3D extends well beyond drug discovery in terms of your concern with high throughput screening, the resulting hypothesis that leads you in number 2. It's very complex, not just 3D drugs.
Okay, I mean, the whole idea is to get a better starting point in discovery, which will lead to development, okay? And in incorporating elements of natural products in drug discovery, you have to be careful because natural products themselves often are not amenable to hit-to-lead and lead optimization chemistry. So the idea is to try to incorporate some downstream concepts into initial design, yet allow for this additional chirality to be incorporated into compounds and really look for the ideal starting point in drug discovery, given one of the things we do is we analyze compound collections that are out and about now, including our own, and say, okay, how can we better represent chemical space, and how can we address maybe gaps that might really give traction against what has previously been considered intractable targets. Okay, anyway, this is all kind of background information leading to the collaborative nature of things here. So anyway, we have quite a few chemical compounds that are on the shelf. We have medicinal chemists and project managers who work with collaborators such as Raj to create compound sets that would be the ideal starting point for drug discovery, and we also do some internal drug discovery too. There's a lot of information here. In short, our 600,000 compounds on the shelf can be divided into a number of parts, but the most relevant part here is our biodesign phase, or the phase in which we incorporate privileged structures from nature into the design of our compounds. And more recently, we have been very active in the area of protein-protein interactions, and we have also undertaken macrocyclic chemistry.
Could you elaborate a little bit on the epigenetics, what you're doing along those lines?
Yeah, I can say we're not quite as active in epigenetics as we are in protein-protein interactions or macrocyclic chemistry, but it has been a relatively hot topic recently, and we're mostly interested in, I could get into some structures and address that in terms of how we get into the different secondary and kind of other areas of alpha helices, et cetera. That would get fairly technical. So anyway, during some break, we can certainly get into this in detail.
Protein-protein interactions, you're also doing that through 3D modeling?
Oh yeah, we have a computational department, certainly. All right, so I'm about to hand things off to Raj. I think you have heard me speak enough, but you can hear more of me during breaks. Again, the whole objective here is, whoops, now I'm going to test the back button. We're coming up to Raj soon. Okay, platform for collaboration. So here's the molecular modeling. Often people have some ideas about a particular library they want to create, but the final compounds aren't on the shelf, so we can take our set of building blocks and possibly source from external sources to create a focused library. We have something from fragments to macrocycles. Fragments might be 300 Daltons, macrocycles might be over 500 Daltons, which kind of goes beyond Lipinski's rule of 5. But anyway, the rule of 5, again, is a dated concept. We do some in vitro screening. We can do some admit testing too, hit confirmation and optimization beyond the initial screen. So there are many things we specialize in providing an integrated set of early drug discovery products and services. And what we do is often look to collaborate. Okay, and I'm going to hand things off now to Raj. Okay, what?
Could you mention any of your successes using this Weber approach, any compounds that you put in clinic, or you have in early phases, or with your partners, of course?
We always try to get feedback from our partners. Unfortunately, they're a bit restricted in the feedback they can give us. It's such that in industry, things get pretty secretive after people generate IP. So our partners who run screens will often tell us, well, the screen went well or the screen didn't go well. Often we sign CDAs too, so this is a pretty controversial aspect of drug discovery in that, if we all worked together and we weren't so secretive, maybe it would be more efficient and we could all go forward better. However, given that IP is the fundamental mechanism under which things work, people get pretty secretive if they have a candidate. So I could, yes, but in the situations where I do have this information, I am restricted under CDA. So it is very interesting, but I'm sorry, I can just share a limited amount of information. Good question.
Here's a living example
Limited of IP or?
of someone you're working with.
Okay, so Raj, I'm going to hand it off to you.
Well, thank you, Mark. So let's move from small molecule to really, really small things, that is bacteria. So I'm basically a bacteriologist by training, and my lab works on this respiratory pathogens belonging to the species Bordetella. And there are currently 9 species of Bordetella, but there are 2 which mainly work on. One is the animal pathogen, Bordetella bronchoseptica. If you have a dog, every year you have to take the dog to the vet for kennel cough vaccine. Bronchoseptica also causes disease in pigs, in cats, and a lot of animals. And mainly, also, we work with Bordetella pertussis. Unlike Bordetella bronchoseptica, Bordetella pertussis is an exclusive human pathogen. It does not infect animals. Humans are the natural species which Bordetella pertussis infects. My lab focuses on 4 areas in terms of bacterial pathogenesis research. One is functional genomics. The genome of Bordetella pertussis, Bordetella bronchoseptica has been sequenced. So we basically, when I'm bored, have nothing to do, I look into the genome and look at new genes and virulence factors. And we have a very good genetic system, and also we have a very good animal model. So we knock out genes and then try to understand their role in different phenotypes, different aspects of Bordetella physiology and virulence. I will tell you in a little bit the vaccines. We are interested in both the animal vaccine and the human vaccine. As you might have heard from a lot of publicity about pertussis, pertussis is the only vaccine preventable bacterial disease that is increasing in numbers in high income countries and in the US. So we are interested in developing efficacious vaccines for pertussis because the current vaccines are not that effective. Bacterial biofilms, this is a phenotype which we got interested in, and these are where bacteria form communities, and these communities become highly antibiotic resistant. So we are trying to understand that. And also the focus of this talk, drug discovery, which we recently got interested in, and that's where we are collaborating with Asenex on this aspect. So like I said, pertussis is increasing in the US. In 2012, there were 48,277 cases and 20 deaths. And this is quite striking, given that there is very high vaccine coverage in the US, the current acellular pertussis vaccine, and still we are not able to control the cases of pertussis. And this was the highest number of recorded cases of pertussis since 1959. There were epidemic in multiple states in 2012, and as recent as this is the data from 2013 or in 2014, and as you can see, there have been an increase in a number of states in the US. I want to point to your attention to California. California currently is going through an epidemic of pertussis. In 2010, they had an epidemic, and 4 years later, they are again having an epidemic of pertussis. So that's where we are interested in vaccine and the drug discovery approach. So by functional genomic screen, being bored a lot, we have been able to identify a number of virulence factors. We have identified an outer membrane protein component, which we are right now trying to develop as a vaccine. We are talking to a number of vaccine companies like GSK and other companies in trying to collaborate with them on this aspect. We have identified a polysaccharide locus, a carbohydrate locus, and an innate immune resistance locus. But for the focus of this talk, I want to focus on this transcriptional regulator BPSR, which is the focus of the drug discovery. So why do we think BPSR is an excellent drug target? We have found that it is an essential gene in Bordetella pertussis. When I mean essential, we cannot knock this gene out. And we have knocked it out only when an external copy of the gene is present. So this gene is essential for the lifestyle or the vitality of Bordetella pertussis. And we have done microarray analysis where we know that it's a global regulator, regulates 64 genes, which have a role in virulence and metabolic and vital cellular processes. And this is what we hypothesize, that this is the reason BPSR is essential. And importantly, from a drug discovery perspective, homologs of BPSR are very highly conserved in Bordetella pertussis strains, including the pertussis strains which were isolated during outbreaks from 2010 to 2012. And this, we think, is important for the broader applicability of any small molecule compounds. And the BPSR family of transcriptional regulators are conserved in a large number of animal, human, and zoonotic pathogens, resulting in the broad applicability to a number of diseases.
Is any of the virulence factor belongs to type 3 effector category?
No, actually, no, none of these belong to the type 3. Many of them are basically the metabolic genes, like in amino acid binding or in the metabolism of the bacteria. So we are researching on what the role of these genes are. So basically, we have identified an unmet need. Other than high-income countries, pertussis still remains a problem, 30 to 50 million cases worldwide and 300,000 deaths per year. And with Asinex, we are optimizing the use of resources. We are using their medical chemistry expertise, their structure-based design expertise in trying to screen a library of compounds for inhibitors of BPSR activity. And I will tell you, just to show you a scheme of what our assay is basically, what we are going to do. So we have a crystal structure of this regulator right now available. It's not published. So we are going to use structure-based design approaches to first narrow down the 600,000 compounds of the library which Asinex has. And we are going to basically screen first for growth inhibition of Bordetella bronchoseptica, that is an animal pathogen, because it is easier to grow Bordetella bronchoseptica than pertussis. Pertussis does not grow at all in 96 well plates, but we can grow bronchoseptica in 96 well plates, and these two bacteria are pretty homologous. And then after we screen in bronchoseptica, we would screen in pertussis, and then we will try to find small molecules which are inhibitory at low concentration, and of course they have to be non-cytotoxic to eukaryotic cells. And then we are going to try to understand how the mechanism of these small molecule compounds, that hopefully they're going to work on trying to inhibit the DNA binding activity of these regulator, because it is a DNA binding regulator, and ultimately we are going to test its effect on the regulated gene expression. So
What are the downstream targets for this regulator?
downstream,
Downstream target, what are the specific targets that this DNA binding protein binds?
so it is regulating about 64 genes in our microarray, and we have actually done assays where we show that this regulator binds to 5 of those promoters because it's a DNA binding regulator. So it binds to the DNA, and then it either activates transcription or represses transcription.
So your microarray data has been validated by different assays?
Yes, we have done independent real-time RT-PCR. I think we now have 10 confirmed targets.
No, I'm not asking you about gene expression assay like RT-PCR, qRT-PCR, and the microarray. I'm talking about the functional assays.
So we have not done, those are pretty highly conserved genes. So generally in bacteria, where you assign the function by annotation, and those are very highly conserved genes. So we have not gone on and actually identified the function of those genes, but since it is essential, I think those genes are going to play a role in metabolism. We are right now focusing on trying to inhibit the activity of this regulator. Yes?
But that regulator seems to be unique to that bacteria, because what other effects would you foresee for a target to that regulator?
So BPSR is conserved in all Bordetella species, and in pertussis it is essential. So, and it is,
But other bacteria.
yeah. So other bacteria, there's a family of regulators. This is a family of regulators called MARAR, and the first member was called multiple antibiotic resistance regulator. And this family is conserved in a lot of bacterial pathogens, and they can either activate transcription or repress transcription. They are involved in antibiotic resistance, they are involved in resistance to chemical compounds, and many of the bacteria, these function as virulence regulators. Like in Yersinia species, Yersinia pestis and Yersinia enterocolitica, there's a homolog called RAW-A, which actually functions as an activator of transcription. So we can talk during the break.
Thank you so much. This is fantastic.